Good day everyone. So today we will be presenting our group assignment and the topic is investigating the mechanisms of gene expression and regulations in plants. I will talk about the introduction of gene expression in plants. The term gene expression can be defined as a process by which the information encoded in a gene is turned into a function. In simpler terms, gene expression acts as on or off switch that control when and where RNA, molecules, and proteins are made. Gene expression is critical in plant growth and adaptation because it decides what traits are expressed in response to environmental cues. Plants use gene expression to regulate processes such as growth, flowering, and defense mechanisms, allowing them to adapt to changing environments including temperature, light, and nutrients availability. Arabidopsis thaliana is commonly used as model plant for the study of modern biology. Research using this plant model is fast as this plant can develop in a short amount of time, from seed to a plant bearing mature seeds in as few as 6 weeks, depending on growth conditions. Studying gene regulation in plants have several challenges, including the complexity of plant genomes, functional redundancy within gene families, and the influence of epigenetic modifications. Recent technology breakthroughs such as high-throughput sequencing and bioinformatics have made gene expression analysis more comprehensive and precise. In this study, the mechanism of gene regulation, the pathway of gene expression, and relevant case studies and application will be further explored.
Assalamualaikum and hi, my name is Muhammad Aif Nakib Wijay Azmi, matric level 78218. Now I will start with my part, gene expression pathways in plants. First is signal transduction pathways that lead to the gene expression. First is receptor activation, and the external signal are detected by receptors like phytochromes and RLK. Then, signals are amplified by molecules like calcium 2+ in second messengers. And then the transcription factors are activated by second messengers, transcription factors that lead to the regulate gene expression. For example, in phytochromes pathway, the light-activated phytochromes move to the nucleus and regulate light-responsive genes. RLK pathway activated by pathogens that lead to the defense gene activation. Next is role of hormones in gene expression. Hormone acts as signaling molecules. They bind to receptors on target cells, triggering signal transduction pathway. There are lots of hormonal effects such as auxin, ghrelin, cytokinin, ethylene, and FCDSC. This hormone has interaction between each other. This hormone has pathway that can communicate with each other for coordinated responses. For example, ABE and ethylene when in stress. This also can lead to feedback mechanism to maintain control of gene expression, such as negative feedback to prevent overactivation, or positive feedback to amplify weak signals.
Hello, my name is Saranya Yukiko, and I will be touching on gene regulation and mechanisms. Gene regulation and mechanisms are processes in which cells are allowed to control the expression of genes and determine their timing as well as level of activation or repression. And gene regulation is vital for cellular development, differentiation, and adaptation to environmental conditions. These key mechanisms include transcription factors and epigenetic modifications. Transcription factors are DNA-binding proteins that control transcription via the recognition of specific sequences within promoters and enhancers. So, generally, there are two types of transcription factors, the first being a general transcription factor, which is required for transcription of all genes, and the second being a specific transcription factor, which binds to unique DNA sequences and regulates very specific genes. And the function of these transcription factors include the enhancement of RNA polymerase interaction with promoters in order to in order to initiate transcription, as well as a repressor in which it blocks DNA polymerase binding or it inhibits its activity, preventing transcription. Finally, I'd like to touch on the epigenetic modification in plants. Now, epigenetic modifications such as DNA methylation and histone modifications play a vital role in the regulation of gene expression in plants. DNA methylation is the addition of methyl groups to cytosine residues, and this actually leads to gene silencing via the prevention of transcription factors binding or the recruitment of chromatin condensed proteins. Histone modifications, on the other hand, include acetylation and methylation, and this actually affects chromatin structure and gene expression.
Gene expression regulation in plants, similar to other organisms, occurs at multiple levels and is crucial for optimizing gene output and coordinating cellular functions. This regulation can be classified into transcriptional and also post-transcriptional pathways. For transcriptional pathways, we will be looking at the promoters and also transcriptional factors. We will also be looking at the enhancers and silencers, epigenetic modifications, and also the chromatin remodeling. For post-transcriptional pathways, we'll be looking at alternative splicing, RNA editing, mRNA stability, and also the translation regulation. So now I'll be talking about the practical applications of gene expression in plants, in agriculture, and also biotechnology. So the first one is crop enhancement. We can observe this by enhancing the crop yield by overexpressing certain genes, for example, the photosynthesis gene. And we can also use gene expression analysis for the selection of better hybrid crops. Next would be the pest and disease resistant. This is observed through the production of transgenic crops, for instance, where these crops have pest or disease resistant genes inculcated in them. And then we could also use RNAi, for example, RNA interference, to help silence certain genes which are harmful pests or also pathogens. Certain genes that are responsible for certain pests and also certain genes that are harmful for certain pathogens, we could silence them using RNAi or RNA interference. So the next one would be nutritional enhancement. So what I mean by this is we try to increase the nutritional content in our products. For example, in golden rice, where we have an enhanced production of beta carotene, and we can also silence certain genes, for example allergenic protein, where we can reduce the production of allergenic protein by using gene silencing. The next one would be in sustainable agriculture. So for instance, we observe this in the case of bioremediation, where we use plants to detoxify polluted soils.